This is the introduction to the tree biology lab on measuring water potential. You recall from class that there are three components of water potential that we consider in trees. And the total water potential at any point in the tree is the sum of three component potentials. Psi pi is the osmotic potential associated with dissolution of solutes in water. Psi p is the pressure potential. That's either negative in xylem elements like tracheids or vessel elements, or positive as turgor pressure in living cells. And then psi g is gravitational potential, just associated with how high up in the tree the particular point you're looking at is. For the purposes of what we are doing in lab today, we make a couple of assumptions. One, we assume that the xylem sap in tracheids and vessel elements that we're looking at is very dilute. It doesn't have many solutes dissolved in it. So we can assume then, we do assume that osmotic potential is zero. For gravitational potential, we either assume that gravitational potential is zero, or we can calculate that just based on height and add that in. So with those gone or accounted for, the water potential of a particular leaf is the same as the pressure potential in the tracheids or vessel elements. And this assumption is what allows us to use an instrument called the pressure chamber to measure water potential of leaves. So the way that works, if we think about a leaf attached to a stem, that's what this is. This is my terrible schematic of a leaf attached through the petiole to the stem of a tree. That leaf is transpiring, so there's a water column going from the stem into the petiole, into the leaf, and then being evaporated from the leaf. Because that evaporation is occurring, there's a tension or a pull on this water column. It's being pulled in this direction. So let's say then that we cut this leaf off. Let's think about what happens to the water column. When we cut that leaf off, because that water column is under tension, is being pulled in this direction, at the point where it's been cut, that water column recedes into the petiole. So what we can then do is take this leaf and put it into an airtight chamber like this, with the petiole sticking up outside the chamber. And then what we do is pressurize that chamber. And by pressurizing the chamber, we basically gradually push that water column back to the point it was when that petiole was cut. So when we first put that cut leaf into the pressure chamber, that water column is receded down into the petiole like this. But as we pressurize the leaf, that water column gets pushed back up. What we do is we observe that cut end of the petiole, and at the moment when we see water appear at that cut end, we read the pressure gauge, and that reading is the amount of pressure that is needed to just counteract the pulling force that existed in the xylem when it was cut. In other words, that positive pressure exactly counteracts the pulling pressure, the negative pressure that was there when the leaf was cut. So we can read the pressure off of this gauge, put a minus sign in front of it, and that is the water potential of this shoot. So there's another video that I made that introduces you to this instrument and shows you how measurements are done with that instrument in more detail. So in this lab, we're going to be using this principle from lecture. So if you remember this principle is that leaf water potential is proportional to several things, proportional to soil water potential minus transpiration rate times hydraulic pathway resistance. For all of what we're doing today, we're holding hydraulic pathway resistance constant, but we're going to vary either transpiration rate or soil water potential and make some hypotheses about some treatments. Okay, so we have three treatments in this lab. These are all branches that have been cut off of trees. In one treatment, I've taken that cut branch and put the cut end into water and then put a plastic bag over the leaves. In another treatment, I have that cut branch in water, but there's no bag over the leaves and I've shined a light on the leaves. And then there's another treatment where I haven't put the branch in water, it doesn't have a bag, and it has light on the leaves. So let's think about what these treatments do relative to this equation. And this equation is important. This is equation number 2 in the lab handout. I'm going to want you to discuss all of the results of this experiment relative to this equation. So let's think about what we're modifying when in these different treatments. So with the cut branch in water, this soil water potential is probably near 0 because essentially that branch is rooted in water. Okay, so with a bag over the leaves, those leaves are in a humid, dark environment. So there's probably little or no transpiration rate. So this transpiration rate is near 0. Okay, so let's think about then what the leaf water potential is in this treatment versus these other two. So let's think about this treatment where the cut branch is in water. So soil water potential is probably still near 0 because it's in water, but there's no bag over the leaves and there's a light on the leaves. So the stomata are open and those leaves are transpiring. So those leaves, this transpiration rate is non-zero. So the question is, thinking about this equation, in this treatment or this treatment, which one should have a more negative water potential? Thinking about how we've modified these two terms. Okay, so I want you to make a hypothesis. And then the third treatment, the branch is not in water. So that's essentially as if soil water potential is more negative. It doesn't have access to water. And the leaves are not in bag and there's a light on them, so they are transpiring. So this is more negative and the leaves are transpiring. What should the water potential of this be relative to this treatment or this treatment? So you're not going to be able to predict the actual numeric water potential, but you should be able to predict the order from less negative to more negative of those three treatments. So I want you to make a hypothesis in your lab report based on equation 2, so discussed relative to equation 2 and what we've modified relative to what you hypothesized to be the order of water potential. And in lab, we're going to measure the water potential of leaves in those three treatments and record them, and you'll record the means of each of those treatments in your lab report. So in the introduction, I want you to spend a little time setting up the lab. Talk about why it might be important to measure water potential, talk about how the pressure chamber works, and make a hypothesis about the pattern of water potential you might expect in those three treatments relative to equation 2. What does equation 2 lead you to hypothesize about those three treatments? I'll post the data set from lab on Canvas. And whether you're an in-person student or an online student, I want you to analyze that data set and present the mean and standard deviations of water potential of the chutes in each of the three treatments in a properly formatted table or graph. Don't present raw data, as always. And then do a results and discussion, describe the results, the patterns, and what they mean. And I want that discussion to be relative to equation 2. So use that to explain what you're seeing. And then I have a couple of additional questions in the lab handout. Either answer those directly or incorporate the answers to those questions in your results and discussion session. As always, contact me if you have any questions as you're writing this up.